Hello everyone and welcome to Longevity Article Review number 6. My name is Jordan Marsh and I am excited that you are here. So this time I wanted to dive into cold plunges. If there was a topic that gave me the best bang for my buck as far as really increasing your chances of living a long life and a long healthy life and a good quality of life, I am not really sure that this is the topic. And so I'm starting with that disclaimer that I recognize that I could probably spend my time on much more important topics, but my goal here is to actually do hundreds, if not thousands, of these reviews. So I feel like I have a lot of time to dive into many different types of topics. And this is one that I've been interested in, largely because I've just been interested in all of the hoopla surrounding it. It seems like just about everyone and their dog says that you need to do cold plunge. You need to do cold plunge. And why? I mean, one reason why I've heard a lot online or on social media from random people is that it reduces inflammation, and that should be good for your body, and it probably is. And so I don't necessarily know that that's true, and after reviewing this article, I'm a little bit even more skeptical of that, but I do want to actually dive into the research myself, and hopefully I can introduce that research to you in a way that actually makes sense. And hopefully we can learn something about it together. And so here we go. So this is the study. We started with 9 physically active young men. What they did was they had these 9 active young men do one bout of single leg exercise on 2 separate days. So what that means is one week they decided, okay, we're going to work out your left leg really hard, and then the next week we're going to work out your right leg really hard. And each session was either followed by cold water immersion or what they called active recovery. And active recovery was basically just riding on a bike. It's kind of like the cool down setting on the treadmill or on a bike that you've seen before. And they were going to test how well either cold water immersion or active recovery influenced inflammatory-like markers. And then they did muscle biopsies from the vastus lateralis of the exercise leg, both before the exercise and after, and use that to also determine inflammatory markers. And so here's an idea of the resistance exercise that they did. 45-degree leg press, single leg squats, knee extensions, walking lunges. All of this took about 45 minutes of exercise. And we did these different interventions both before and after. So let's talk about exactly what they were. So for the cold water immersion, they sat for 10 minutes with both legs immersed in water up to the waist. And then the temperature was around 10 degrees Celsius or about 50 degrees Fahrenheit. The recovery therapy was just 10 minutes of self-selected low-intensity work on a stationary bike. And the mean power output for each of these individuals was around 37 watts. Okay? So kind of an interesting comparison. We are comparing cold water immersion to active recovery to see how well they influence inflammatory markers. And then I wanted to give you a really succinct idea of just what they did with the blood samples. So first, they took a blood sample before the exercise. They did it immediately after, which I think is important to know exactly what happens before there's even any type of recovery therapy. They did 30 minutes after, 1 hour after, 2 hours after, 24 hours after, and 48 hours after. I should say that it was about 5 minutes after the exercise that they engaged in the therapy. So 5 minutes after the exercise, well, first they got their blood sample, and then 5 minutes after they either hopped on into a cold plunge or they did some recovery therapy, okay? And then all of the times afterwards were times after the exercise, knowing that we had undergone the therapy. Similar thing for muscle tissue samples. They took one before exercise, they did one 2 hours after, 24 hours after, and then 48 hours after. And one thing that actually I thought was pretty interesting was that the before exercise and 2 hour after muscle tissue samples or muscle biopsies were done in the same incision, but they were worried about, inflammatory markers being there just because there was an incision. And so what happened was they pointed the incision in one way before the exercise, then they pointed the incision in a different way in the next exercise. And then at 24 hours after and at 48 hours, they made new incisions to try and ensure that they weren't just getting noisy inflammation. So I thought that was interesting. There's many different control type procedures that they underwent. And I'm not going to go through all of these in depth, but just know they really worked hard to ensure that the types of meals they were eating and the supplements that they had and the protein that they were drinking was very similar for each of these 9 subjects. Okay, so here's some results. I don't want you to get too freaked out by it, but let's go through it just really briefly. So each of these graphs show a very similar x and y axis. So on the x-axis, you see here up at the top left that 2 hours after exercise, 24 hours after exercise, and 48 hours after exercise, we are measuring something. And then what it says up here is the fold change, or the change in that particular marker compared to the pre-exercise value. Does that make sense? So based on the pre-exercise value, we're seeing how much that marker has changed 2 hours after exercise, 24 hours after, and 48 hours after. And we are doing it for ACT, or active recovery, and in black for cold water immersion. Okay? So let's just say, and for all of these, I have kind of tried to make it clear here that these are just markers of inflammation. I don't want you to get too concerned about exactly which types they are. Just know that they are markers of inflammation that we're looking at. So if you think about this, you would think, okay, let's say cold water immersion is the king of reducing inflammation after exercise. You would expect in these graphs that there should be no significant difference between the pre-exercise level of these inflammatory markers and the post-exercise level of these inflammatory markers. And you would expect that active recovery probably is not as good at calming down inflammation as cold water immersion, if it is as good as we think it is at reducing inflammation. So what I'm saying is, for each of the black dots on these graphs, you should probably expect to see no stars next to them, okay? And for each of the white dots on the graph, you should expect to see more stars. You should expect to see stars because you would think the inflammation is still high. We haven't treated our body with cold to reduce the inflammation. And we think that, it should be high. It should be high relative to the pre-exercise value. So I hope that helps to kind of see what are we looking for when we think markers of inflammation. Should cold water immersion, kind of as a hypothesis, should be able to reduce inflammation much better than active recovery. And as you can see, you don't necessarily see that here. So let's just start with neutrophils, for example. So yes, there is a significant difference with active recovery at 2 hours based off of the pre-exercise level, and that there are not any stars next to cold water immersion in neutrophils. So I guess that checks out. We expect there to be no inflammation with cold water immersion and more inflammation with active recovery, and that's somewhat true for neutrophils according to this graph. But that's not necessarily the case, say, for example, with MAC1. MAC1, yes, there's more stars associated with active recovery, but there's still a significant change in inflammation with MAC1 at 48 hours for cold water immersion, right here. So we know that didn't necessarily pan out exactly. How about CD163? Same story. Here's a 24-hour value for cold water immersion that is significantly different. It's interesting. So yes, maybe cold water immersion isn't going to be perfect at reducing inflammatory markers, but it's also not necessarily passing this eye test to me of being able to do that. Here's a very similar graph for both cytokines and chemokines. And cytokines and chemokines, I wanted to say exactly what they said in the study about these and why they were investigating these. So in the present study, we focused on changes in the gene expression of all of these different markers because these cytokines are responsive to mechanical loading associated with exercise, and they play an important role in recruiting inflammatory cells. So they're recruiting inflammatory cells, obviously part of inflammation, to damage muscle tissue and regulating muscle repair. So these are the tissues that should be regulating our muscle repair. Kind of the same thing as those inflammatory markers in the last slide. We should expect cold water immersion to quiet down these markers of inflammation. And it's not necessarily the case that it does. So here, even after cold water immersion, we have a significant change. Here, we have a significant change. Here, it's significant across the board. And for IL-6, this one was fascinating to me. For IL-6 right here, it was active recovery that was actually better at reducing inflammation. So here it's significantly different, but here it isn't, which is like, wait, what? But cold water immersion stayed high. So are we sure that this is actually reducing inflammation like we think it is? It's up to debate. They did a bunch of other ones. These are more cytokines and chemokines. Again, I'm not going to go too far into exactly what they are, but I'm just going to highlight that there were some significant changes in inflammatory markers for cold water immersion. And we're showing that active recovery is sometimes even better than cold water immersion at quieting down some of these inflammatory markers. Here is a graph of neurotrophins. So neurotrophins are a family of proteins that are critical in neuronal development or neuron development and growth. And I thought this was interesting because they specifically chose these markers because they're associated with muscle soreness after exercise, which is interesting because we all know, at least I've heard a lot about how cold water immersion or cold plunges can reduce the feeling of muscle soreness. And so if these are associated with muscle soreness, I would expect that cold water immersion would tamper them down quite a bit. So here, GDNF, you can see that there is no real difference. So here, there's a significant change in GDNF with both active recovery and cold water immersion, but no significant change here and here. So active recovery and cold water immersion are about the same. And again, NGF, with cold water immersion, we actually see that it is worse than active recovery in reducing that neurotrophin. So I don't know. It's a little bit, I'm a little skeptical. And here, heat shock proteins. Heat shock proteins are commonly, they are overexpressed in times of stress or challenge or heat or exercise. And so their role is to regulate proteins that are under stress and maintain them as best they can while they are under stress. And so you would expect that after exercise, they would be upregulated, which is the case. And again, both active recovery and cold water immersion show that these come down after 24 hours and 48 hours. So I don't know. What can this tell you? It's very interesting to me that there seems like there's a theme that there's not really a difference between active water or active recovery versus cold water immersion. So here's 3 good things that I got from this, and I've kind of hopefully alluded to this as I've gone on. So these findings provide evidence against the traditional notion that cryotherapy, such as cold water immersion, helps to restrict inflammation and cellular stress responses in muscle following exercise. I've probably said that upwards of 5 times already. There's this traditional notion that it really helps with inflammation and cellular stress, and that's not necessarily what we found here. These findings suggest that cryotherapy does not substantially alter local or systemic inflammatory responses to exercise-induced muscle damage. So remember, there's a local response. We're doing a muscle biopsy for some of these markers, and there's a systemic response. We're doing blood tests for some of these markers. So both locally and systemically, there's not really a difference. And then they specifically mentioned that less muscle soreness after exercise seems to be the only consistent effect of cold water immersion. That was kind of interesting to me that they specifically mentioned that, because the neurotrophins that they specifically said were part of, they were associated with muscle soreness were not necessarily brought down by cold water immersion. But again, it seems like that seems to be the thing that most people talk about as a benefit of cold plunging, is reducing that feeling of soreness after you exercise. And so I think I've heard this before, that regular long-term cold water immersion might actually be detrimental for developing muscle strength and hypertrophy, because these inflammatory cells are actually supposed to help in muscle repair and building up your muscles. but they may assist occasionally on helping you recover faster and not feel so sore. But that might not always be a good thing based off of what your goals are. So here's 3 things to consider. This was, again, a small study of 9 subjects, only men. They actually mentioned something I thought was interesting. 6 of the subjects performed their second round of single leg exercises 1 week after they did their first round, and 3 of them actually did it 4 weeks after, just kind of due to some kind of scheduling issue. So something to think about as to why maybe some of these results may be skewed. And some studies suggest that adaptations can happen in a contralateral limb when you go through single leg exercise. So they made efforts to address this, but still a consideration that other studies were actually showing that you may undergo significant changes in one group of muscles on the contralateral leg, and that can skew some of these results when you go through that exercise again a week later. So thanks for coming, but again, I will also get into many other different topics that are likely more important for your chances of living a long and healthy life. All right.